Good morning. Let me begin by expressing my gratitude to the gracious host and all the distinguished guests gathered here. My name is YongKeun, and I'd like to talk about holotomography system, the principle and application, and how it can be used for phenotypic drug screening. So to make a long story short, all holotomography or HCT is an optical technique to visualize three-dimensional shape of the cell with high resolution without introducing any exogenous labeling agent. So you can see the live cell without use of the fluorescent proteins or staining dyes, and you can see all the major subcellular organelles, such as you can see here. You can see the chromosome condensation, alignment, separation of the cancer cell line, and those sparkling, the little spheres are lipid drumlet. You can also see nucleus membrane and the heterogeneity in nucleoplasm. And the question is why we are doing this, why we are developing this HCT system. So currently, the driving force for the molecular cell biologists is based on the use of fluorescent protein, which is good for the molecular specificity. But I see that for the next decade, there are going to be a lot of the research work to be done in the field of 3D biologies, including organoids and microphysiological system, and also the regenerative medicine, such as the use of the iPSC colonies, therapeutic agents, and etc. So for those applications, we need 3D imaging capability and without the use of the exogenous labeling agent. HCT is an optical analog to X-ray CT. In CT, it measures multiple 2D projections of X-ray images as a function of various illumination angle, from which you can reconstruct three-dimensional distribution of X-ray reflectivity without having any invasive process. In HCT, it measures multiple 2D hologram using light, and it measures multiple 2D hologram as a function of illumination angles, and then it reconstructs three-dimensional distribution of the refractive index of live cells and tissues and organoids. And when you use HCT, you can overcome several challenges and limitations of conventional imaging technique. So no preparation is required, including fixation, staining, or the labeling. And when you use a fluorescent microscopic technique, it requires a sample preparation as well as due to photobleaching and phototoxicity. Even though you may generate beautiful fluorescent images, but you have to discard because cells are not viable for a long period of time. And for 3D imaging, such as organoids, you have to do fixation, staining, and antibody staining, and measuring the images. But this is a still shot of that organoids. And obviously, you cannot use those organoids for therapeutic application. But using HCT, no preparation is required. So by simply taking out multiple plates from your incubator, place in our HCT system, and click a button, it takes 3 seconds to measure 1 tomogram. And then we also have incubator inside, so you can just keep watching the cell without having any external artifacts. And we can measure the 3D samples up to 150 micrometer, so you can keep growing, measuring and growing organoids for a long period of time, and also you can use for the therapeutic application. Those are key benefits of using HCT: no labeling, and thus low phototoxicity and photobleaching. And we can measure for unlimited time, and our speed of measuring is about 100 times faster than confocal microscope. And our resolution goes down to 150 nanometer, and we can measure the sample up to 150 micrometer. I'll talk about quantitativeness in the following slides. So this is a screenshot. This is still the one slice of the tomogram at a specific actual positions of cancer cell line. And you can see cell membrane and nucleus membrane, nuclear line, mitochondria, lipid drumlet, and even see the vascular structures over here. And unlike conventional phase contrast microscope, label-free technique, we have high resolution 3D and quantitative information. And without using any exogenous labeling agent or fluorescent proteins, we can visualize major subcellular organelles. So in comparison with conventional fluorescent confocal microscope, this is mitochondria measured by the fluorescent microscope technique. But if you use the same cell using HET, you can see this cell over here and good correlation over mitochondrial area between the fluorescence and our refractive index distribution. And you can also see there are adjacent cell, which is not seen in the conventional fluorescent microscope, maybe due to the low expression level, but we do not miss those information. And this is the image that is shown in the title slides. And since we have no labeling requirements, so you can see very sensitive cell. So this is primary cultured neuron cell. You can see transportation between neurons and dendrite structures, as well as the subcellular organelles. And if you watch the cell over here, it's trying to make a new connection over here. So it's trying to search and suddenly changes the direction and go to the cell and making a new dendrite structure here. So this is 12 hours movies without having any difficult procedures. And for those who are studying the metabolism and lipidomics, they are interested in measuring the dynamics of mitochondria, fission and fusion, together with other major subcellular organelles, such as the nucleus membrane, nucleoli, and lipid channel. Those are clearly seen over unlimited time of the measurements. When you say long-term imaging, it's not hours or days, it's weeks and months. So this is same field of view of fibroblasts differentiating into the adipocytes over 42 days, and we are watching how those cells are undergoing the alteration in terms of morphological features. And you can also watch the lipid drumlet generation, which is hallmark of the adipocytes. And this is eventually actually the tissue-like structures, but you can see those images over a long period of time. And we can also cover larger field of view. So by measuring the individual field of view or region of interest and stitching them into the large size data. But this is 4 millimeter by 4 millimeter tomogram of the adipocytes of the days of 42. And you can see the subcellular organelles as well as the those lipid drumlet. And like Google Maps, you can just go back and forth and covering this large field of view. Can you guess what is this samples? This is embryo, mouse embryo at the stage of 2 cells. And you can see this, clearly see the 2 cells here, and those are surrounding sperm cell. Size of this embryo is about 150 micrometer, but it takes only one shot. Sorry about that. It only takes 3 seconds to measure this one volume of the data without having any the use of the exogenous labeling agent. And you can also keep watching how they are differentiating into the blastocysts. And with the collaboration with some of the hospitals in South Korea, we are developing the AI networks to predict which early stage of the embryo will go into the expanded blastocytes by using the machine learning approaches. And this is iPSC colonies. And if you use bright field microscope, it generates only 2D and low contrast information. But using HET, it has high contrast and also provides volumetric imaging capability. So you can see the multiple layers of the cell in the volumetric representation. We can also measure sick tissues. So this is the mouse brain. But fixation is optional. You can also measure fresh tissues because we do not use any labeling agent. But the important thing is the thickness is about 150 micrometer. In conventional histopathological practice, they do fixation and sectioning, like followed by the some of the embedding, stuff like that. And typical slide thickness is about 3 to 5 micrometer, but in this case, 150 micrometer. And then you can see the various structures like that. This is the hippocampus lesions. You can see dendrite and the individual cell and other structures in 3D. This is organoids, mouse liver organoids, and it has a shape of a shell But for visualization purpose, we only visualize the bottom half. You can see individual cells and their subcellular organelles, such as the nucleus and nucleoli and lipid drumlet. But important thing is this is live and no protocol changes. Live organoids in matrigel, and this is the same protocol that people just grow the organoids, and you're watching how they the individual cells are dividing and undergoing cell death, including apoptosis and necrosis. And we can also measure for a long period of time, and this is small intestinal organoids from mouse from the size of 15 micrometer, and it grows until the size of about 200 micrometer. And you can also see the lumen structures and individual cell. And this is how we are measuring over 7 days. So you can just keep watching while these organoids are growing in your microscope, holotomography microscope. Okay, so one of the features of using HET is quantitativeness. What I mean by that, the only information that we reconstruct is the refractive index distribution. So, but even though I haven't shown in the grayscale in the previous slides, there we have the value of refractive index, measured and precisely reproduced. And there is linear relationship between the refractive index value and biomolecular concentration in the units of gram per liters, which means if you measure 3D distribution of refractive index, and you can retrieve the local concentrations of the subcellular organelles. So for example, this is hepatocyte measured by using holotomography, and those are lipid drumlet. And if you release the value of refractive index, you can calculate what is the concentration of the specific lipids in the unit of gram per liters. And again, this is volumetric imaging system. So you can quantify the volume corresponding to the specific lipid drumlet in the unit of femtoliters. And now you have concentration and volume. And if you multiply those two quantities, you can calculate dry mass of a lipid drumlet or other subcellular organelles without distorting the cell, without having introducing exogenous labeling agent. This is highly quantitative because of this linearity between the refractive index and concentration. And when it comes to the bioimaging for the study of the cell biologies and the drug screening and also for various regions, I think those 6 parameters are all important. One is spatial resolution, how small you can distinguish between two objects. Temporal resolution, how fast you can measure this phenomenon. How deep you can measure the sample. And SNR, how strongly you can distinguish one signal from the other one. And observation duration and sample health. For example, fluorescent confocal microscope is great technique, has been used for the decades, but because of its high SNR, because of the use of the fluorescent protein, but it has some limitation in spatial resolution and temporal resolution and the sample health and observation duration. Even though we are not using fluorescence, we provide the totally different information about the morphological phenotypic information. And we can distinguish major subcellular organelles, and our spatial resolution is about 150 micrometers, slightly better than confocal microscope. Our temporal resolution is 100 times faster than the confocal microscope. And at the same condition, we can visualize more thicker samples than confocal microscope. If you recall some organoids images measured by confocal microscope, they do fixation, clearing, index matching, and etc. The same condition, holotomography can see better than confocal microscope. But the most important features that holotomography can provide is the sample health and observation duration, because we are using intrinsic imaging contrast, effectiveness, no preparation, no fixation, no staining, and those are the key benefits of using HET. So, for years ago, a few years ago, we have founded a company to commercialize this system, and this is one of the systems that we commercialize and on the market, and it can measure both HT data as well as the multi-channel 3D fluorescence. So you can perform correlative imaging For example, it's like PET-CT. The PET provides, in medical imaging, PET provides molecular specific information, and CT provides structural information. And we do the similar thing. HT provides the overall the morphological information of the cell, and 3D fluorescence can provide the molecular specific information. And together we can do the better job. So those are the early adapters, and those are the papers from our users. And for the sake of time, I do not have, and I cannot go into the details of each application. But the major application field is the organoids and the regenerative medicine, such as iPSC colonies. And phage-phage separation is also good application, and the cell biology, etc. And from the last part, we have talked about the commercialization part and the principle of the HT, but this is an ongoing project. And we are trying to find the better applications of HT, and we realize the use of the machine learning and AI is crucial because we generate the new type of information about the cell structures, and our data size is huge because it has we have high resolution and the 3D imaging capability, and also for the long-term period of imaging. So we have developed a series of the technique for segmentations of the cell and the nuclear organelles. We have developed the AI technique to classify the cell type and virtual staining. But today, for the sake of time, I just touched the two applications. One is the AI-enabled the automatic segmentation, which is about to be commercialized, and we are launching the software in the next few weeks. And by clicking a button, it automatically generates the cell membrane and nucleus membrane and mitochondria altogether from the HD data measured in the stage of the low labeling condition. And second application of HT is virtual or digital staining of the fluorescent markers without using fluorescent markers. Sorry about that. So one of the strengths, the approach of using HT is label-free, and we can provide some major subcellular organelles without using exogenous labeling agent. But one of the limitations is that we have limited molecular specificity. That's why many of our users are actually prepared the fluorescently labeled cell, and they measure HD data, and consequently they measure 3D fluorescence for qualitative imaging analysis. But in the end, we want to develop the technique to visualize and retrieve both structural information as well as the molecular specific information without using labeling agent. So what we have done is we relied on the use of the GAN networks, and for training purpose, we have personally labeled the cell, and for the same cell, we have measured the HD data, and then we measured the 3D fluorescence, and we have trained the AI networks for this paired data set. And once training is done, you don't need to label the cell anymore. And by measuring the label-free holotomography system of the cell, the images of the cell, and clicking a button, and you can generate the virtual 3D fluorescence. And accuracy was very high, more than or greater than 97%. And left-hand side, and you can see the label-free HD data. And the right-hand side, this is all virtual 3D fluorescence in 3D. And you can do 3D biology analysis, spatial biology analysis. So this is the input data set, and those are the all AI has generated. So as of now, we are working on the development of the phenotypic drug screening method based on the use of the label-free holotomography system and the use of the AI approaches. So with that, I'd like to thank for your attention. And this work was done by the collaboration between the Tomocube and KAIS, and I'll be happy to answer to your question. Thank you for your listening.